Our research is in the area of molecular diagnostics and our new variation on the polymerase chain reaction, or PCR. And I'm presenting this work on behalf of our research team and also on behalf of our spin-off company, Monoquant, which has been formed to commercialise any IP that comes out of our group. Molecular diagnostics is that area of laboratory diagnosis which is based upon the detection and study of DNA or its related molecule, RNA. It has an enormous variety of uses. In clinical practice, the most common area is in relationship to infectious disease. And virtually every infection known to man can be detected and or monitored in molecular diagnosis. For example, it's sad but true that the commonest, in terms of bulk, the commonest application of molecular diagnosis is to detect sexually transmitted diseases. But it's also, molecular diagnosis is a key part of treatment of HIV or AIDS, malaria, TB, influenza, swine flu, Hendra virus, any infection, you name it, and there's a molecular diagnosis test. However, molecular diagnosis is important in other areas. Cancer, increasing use in cancer, in inherited genetic disorders, and really in every branch of medicine. But also molecular detection and diagnosis is important in agriculture and even in things such as study of water or soil. Now, the polymerase chain reaction, or PCR, is a fundamental technique. It's the most commonly used technique in molecular diagnostics. It's so important that its originator, Kary Mullis, was awarded the Nobel Prize in 1993, and it has revolutionised molecular techniques. In practical commercial terms, the market for molecular diagnostics is growing at about 20% per year, and it's estimated that the worldwide market will be approximately $15 billion by 2015. We have actually been using molecular diagnostics, particularly in the cancer area, particularly in detection of leukemic cells to monitor treatment, and also in diagnosis. Now, what is PCR? What is the polymerase chain reaction? Essentially, it boils down to detecting a needle in a haystack, a rare target. The needle is a molecule of DNA that is of interest. And the problem is that that needle, that molecular target of DNA, is hidden within perhaps 10 million or 100 million other DNA molecules. How does the PCR detect this molecule? It uses two small bits of DNA termed primers, and these are synthesized so that they are a mirror image of small bits of the DNA target. And they go in, they bind to the DNA target specifically, pull it out, and it's able to be amplified out of that great big haystack of multiple other bits of DNA. In most cases, this technique works very well, but sometimes it doesn't work quite as well because the primers also pull out a certain amount of rubbish, and this can obscure getting the target that you want. A common way of overcoming this problem is to perform the PCR twice. You perform one PCR, open the tube, take an aliquot out, put it into another tube with a different set of primers. In a sense, then you have a much smaller haystack with your needle partly amplified, and then perform a second round of PCR. So by the use of two pairs of primers, you get increased specificity and you can pull out your target. We have been using this two-step approach for many years, about 15 years, for our work on sensitive detection of leukemic cells. But recognizing the problem of this two-stage technique, and it has two major problems. The first one, it's more complex technically, but more importantly, when you open the tube, there's the possibility of contaminating the environment. If that happens, that can be an absolute disaster because the amplified target can contaminate further reactions. So we devised this technique termed antisense PCR. It involves the use of two additional molecules added to the PCR, and these molecules have the ability, depending on their structure and design, to either switch the primers off or switch them on. And so we developed this antisense PCR so that the two-step PCR occurs in the one closed system. For the first part of the reaction, the first pair of primers works, it's then switched off, and then the second pair of primers kicks in and finishes off the reaction. Now, we have demonstrated proof of principle of this technique in 4 model systems, and the work has been published. The next phase of our work, and that has both practical clinical use and also commercial potential, is to develop a number of practical clinical uses for this development of antisense PCR, particularly in the infectious diseases area. And if this is successful, we hope to commercialise this through our sister company, Monoquant. Now, why should you invest in Monoquant if some of the audience is investors? Well, there are a number of good reasons, we think. First of all, there's a close collaboration between the scientific group and Monoquant. We as a scientific group have a track record of developing a number of practical and useful laboratory techniques. Our techniques that we've developed for diagnosis of lymphoma and following the progress of treatment in leukemia are now widely used throughout the world in many laboratories. Monoquant currently has an IP portfolio of 6 patents which are at various stages in the process. The initial patent of the present portfolio has actually been awarded now in the US, Mexico and India in March of this year, and other other countries it's still going through. Of the other 5 patents, 2 of them are specifically related to antisense PCR. We have a licensing record. From our work, there have been 2 licenses signed. Most recently, Monoquant signed a license for its hematology IP with the US company InVivoScribe based in San Diego. And the first sub-license seems to be, the signing appears to be imminent, signing with the Mayo Clinic. In terms of cash flow, at the present time, we have enough cash in the kitty, beg your pardon, to enable us to keep going for another 2 years until the end of 2013. We anticipate, however, that we'll need about 4 years to develop our portfolio of clinically useful applications of antisense PCR. Hopefully, some royalty income from InVivoScribe will help to meet that gap. The individuals in our research group are scientifically committed. They're also, some of them are financially committed as they also hold equity in Monoquant. Importantly, of course, we believe we have an excellent product, antisense PCR. The potential market is very large. PCR-based molecular diagnosis will amount to about 15 billion. Obviously, though, we're only looking at capturing a fairly small portion of that market. It's unlikely we're going to hit a major winner, but it's always possible. But even a small slice of such a large market will still be quite large in absolute terms. So what are we asking for the investor community? We think that the sum of $400,000 for 2 years will enable us to keep going for another 2 years till the end of 2015, and this will enable us to develop a portfolio of useful clinical and thus commercially useful applications of antisense PCR. Thank you.